Welcome to Learning With Lowell. Today we're joined with Andy Lee, who is the co-founder of Vincere Biosciences, which seeks to bridge the gap between AI and biology and conquer age-related diseases. Andy, welcome to the show.
Thanks. Great to be here, Lowell.
I was digging through your Twitter, and just for everyone noting in, Andy was one of the first people that pinged his social as well for questions. So anyone listening, if you ever have questions, like you can write in and actually get it read out. And there's one person who actually made a comment they're going to be getting that asked later. But on Andy's Twitter, you made this post saying, why is it that I always end up talking about bio at AI events and AI at bio at AI events and AI at bio events? I jarbled that. How does that happen? Can you just walk through that actual scenario? Because when I read that, I just started laughing.
Yeah, I think it's I think it's somewhat unusual to be bilingual across these disciplines. And so I go to both quite a bit. And somewhat, I think it's, people are always interested in what they don't know and how it can be applied to the areas that they are interested in. And so, I was actually we went down to speak at UAB last week. And again, I gave a talk on how to use big data for drug discovery and neurodegeneration. But the key at the social afterwards, I kind of talked about some of the emerging things with ChatGPT and LLMs, and that was the area everybody wanted to talk about. It's kind of interesting to chat through those extra areas of the computation stuff with the AI people. When I go to the AI conferences, they're all interested in longevity and what molecules are actually working or emerging to the clinic, or how we go about drug discovery. So I end up just talking about those, and I think it's that curiosity on both sides. People want to know about what they don't know.
Mm-hmm. And then you have the company that bridges the gap and can do both. And so for them, for either party, they can just follow what you're doing and get a little bit of both at the same time, which is kind of fun. So a lot of people say AI, but it means different things. What does AI mean in conjunction with what you guys are building?
Yeah, definitely. It's an interesting question that we've been debating for a very long time. I think, to my mind, AI is any computational system that can make new predictions off of data, if it's not explicitly coded to do a thing, but as it gets more data, it can refine. And so by that definition, a statistical regression is a form of AI. It can get better as it gets more data. I know a lot of people now are, something that appears to be of human intelligence, which is, I think, that gets into a whole lot of questions around consciousness and human intelligence, and where do you draw the line between human intelligence and cat intelligence and plant intelligence, all of which have some variety of, I sense new information in my surroundings and decide to have some outcome that will make me feel better or avoid the danger or find the food. So where that line is, and there are all kinds of computational systems that have that level of sensors of their surroundings that they can make decisions on and get better as they go. So I think I use the term very broadly in going from, and it depends on who I'm talking to. And in some cases, I avoid the term altogether and just say we do data-driven drug discovery. In some cases, it's big data. Sometimes it's computational or machine learning. But I think all of those are in some ways synonyms, especially when you look back to some of these algorithms were, defined in the 60s and earlier, and are really now being used. Right?
Do you develop an algorithm for what you're building, or do you take off the shelf algorithms that exist, like open source ones, and apply it to what you're doing and then train it off of your data? What does that look like?
All of the above. So our most complex system is a biological simulation platform that we have a US patent around, where we wrote C CUDA kernels to do a new type of physics-based computation to simulate the biological interactions inside of a cellular system, all the way up to traditional convolutional neural networks, where we take a bunch of methylation or transcriptomic or image data and build a multi-layered neural net that will train on that data, and you do a train test, and now you have something that you can do predictions on naive data, through to custom chatbot integrations where I'm just calling libraries from OpenAI or Huggingface. And so have kind of the whole gambit there of what might be called AI.
Yeah, I recently discovered Huggingface, and I'm surprised at the amount of information that people have freely given out on there. Like there's like little versions of every closed system that people, well, I guess they're reverse engineering it, but it has the same functionality as like some of these closed systems that people haven't opened up yet. It's pretty powerful stuff on there.
It is, and in a lot of cases, the open source algorithm on Huggingface is actually at the heart of what are the popular closed systems that are available. I mean, it's been really predates a lot of what the current wave of AI has been.
Do you think so? I think I was recently listening to a conversation with Mark Zuckerberg, and he was saying that he thinks that the short term is going to be one on closed systems because they'll be able to, hide or whatever, obfuscate, and they'll be like really well trained on specific things. But that over the long term, over the next like 5, 10, 15 years or even longer, that open source systems are going to do better. Do you, as someone who's more knowledgeable on this, how do you see that dichotomy playing out? Do you see a closed system where, and even then, like, to what extent is it an open source system with anything that Facebook does? Do they have their weights and measures opened as well? I haven't looked into their algorithms and stuff to know that, and I don't know if I would appreciate it if I was looking at it anyway, because I don't have the knowledge base. But how do you see the future running on if you look over the next 5, 10 years, what models do you think are going to be more powerful? The ones that are closed right now, or the ones that are open, trying to be more open?
I think you'll end up with both emerging into this kind of battlefield of options, kind of similar to iOS and Android, where technically Android is open, and you could go and take that and build your own version. Linux out there floating around. I think similarly you'll find most work being done with the closed systems, but a good chunk of open being there kind of as a safety net, because there are a chunk of the world who want to make sure that there's an open option. I'm glad that there are. I will say Facebook in particular has been a really good player in the open AI, in the open source AI world, contributing a lot of the underlying libraries that drive AI. And the Facebook AI research group has put out some really amazing research that have pushed the ball forward. Some of it, on par or better to what OpenAI has been doing. They just didn't put the marketing push on it because they were targeting toward research and development teams. Whereas, and even OpenAI, I'm not sure they really understood or expected the massive adoption they were going to get when this launched.
Yeah, and I'm a bit. I kind of wish they would change their name, because I think they're becoming more like ClosedAI with some things, with occasional things being closed. Open.
Yeah, it's effectively Microsoft AI at this point.
Okay, yeah, that's what I keep hearing. I watch these interviews with Mark or other people like Elon, and Elon in particular is quite salty over this, considering he helped found it, and he apparently got the lead guy in there to come in. He poses this question: how can something open source that's a nonprofit turn into like a business closed source thing? But I don't understand that. Do you have like a theory on this, like how that was able to work without people getting in trouble or whatever, like going from something that's open source to closed like that?
Yeah, I think so. From a corporate structure perspective, you see this all the time, where a private for-profit company will make a nonprofit subsidiary, or make a nonprofit that then they can move money through or raise capital for philanthropic purposes, and and the other direction, where you have nonprofits that create a for-profit subsidiary and put things within that they can then have profitable ventures around. In some ways, I think R&D and science should be profitable if you create value in the world. And clearly, OpenAI is creating value in the world. It's unlocked a lot of new capabilities, and people are building new businesses around them. So it makes sense that there could be profit to be had there. Some of the tooling is still being made available for open use, if not open source. And a lot of it is built on the same open source libraries underlying that, like, you could theoretically train your own model if you had your own data set and do the same thing. I don't know that they should have to share their weights, though.
What is the significance of sharing and not sharing their weights? Is that like the secret sauce when it comes to these things?
I don't even know if the way neural nets work, if the weightings of the nodes would be reverse engineerable or reproducible. I suppose the one nice thing is you would be able to take that, host a local version, and then fine tune it with new data to some other purpose, which is not currently, to my knowledge, available with OpenAI as the starting point, at least. Whereas it is with some of the underlying, like GPTs that you can find on Huggingface.
I was watching this video of a person commenting on a video with Mark, where it was like an AI engineer, just like sometimes Mark Zuckerberg will say things in a vague way because that's just how business people do things, and he was like, this is what it actually would look like, the science of it, and they were saying that the reason they're more open source is because people can train the data on things that maybe Facebook doesn't want on their balance sheet. So they can, like, there can be some like kind of more far out experiments going on. I guess like one may be like, like OnlyFans type stuff where like it's really hard to get funding and make business people happy with that for some reason. I think like I read something a couple of years ago where OnlyFans almost went out of business because of the type of content on their site. And so maybe like that type of stuff or related type of stuff that doesn't let their investors be happy or their corporate earnings be happy is another benefit of letting it be open source, because then other people can be taking that risk and you still be learning from it because it's open source. Like I imagine some data is coming back in terms of what's working, what's not to Facebook and stuff from that.
Yeah, absolutely. And that's where those underlying libraries being open is really important, because I think, even getting into more mainstream businesses, drug discovery or banking type things where you've got a lot of IP or really sensitive information that you don't want out, you can't run those through a third-party system for training, but you could host an on-premise, local, or even on cloud on a private set instance of those and train your own model to have those in. So I think that's going to be the really interesting next wave of implementations, certainly through the enterprise. The banks aren't going to implement OpenAI. They're going to build their own private instance. The banks are probably going to hire Microsoft, who's going to use OpenAI to then make a private instance that does extend off of those, just because of those pre-existing relationships. But you can go right now to Huggingface or to the underlying libraries there and build your own GPT or LLM transformers. Like, that's all open source and available to throw it at your own data set, build your own tokenizer, and like from scratch in a disconnected setup, you can reproduce all of those. And a lot of that is because Facebook has made those tools available.
So I'm still pretty new to Hugging Face, and maybe people listening are like, why do they keep talking about Hugging Faces? But Hugging Faces, could you just briefly define it, and then where do you go on it? Are there like certain watering holes in particular? Maybe it's all like GitHub, so maybe there aren't watering holes, but I still think that there are watering holes on GitHub where people consolidate information and then make link trees essentially. But yeah, so what is Hugging Face, and then how do you use it?
Yeah, so Huggingface is an open source or a repository of open source LLM AI models and tools. And it's pretty cool in that most of them have a demo page right there where you can drop in text and run a query and like test the things out and see what the responses would be. And the APIs are very clearly documented and easy to implement. And with that, you can include a few libraries and download a starter model and then fine tune it to a new purpose and then start to run your own local LLMs using that. And it's become a very big repository of different types of AI models, everything from image generation, music generation to chatbots and GPT type things. Some of the ones I've been playing with are like BioBert, which is another LLM trained exclusively on biological data, like from PubMed. And so it achieves higher accuracy on scientific text generation and interpretation as opposed to like ChatGPT that's trained on everything. And then you get into, there are other pros and cons depending on which one, but it's pretty easy to go and browse. So if you're interested in building your own AI stuff, I would encourage to take a look at Huggingface as a place to get it. And they're all implementable with Python code. And if you want to get started, ask ChatGPT to generate you a Python script that will call the Huggingface APIs to train a custom model. And you'll get a pretty decent starting point that you can then just run in any interpreter.
Yeah, that's the weird thing. It's almost getting like paint by numbers a little bit. When I ask ChatGPT for code, like it's like most of the time it's right, or at the very least it'll be wrong in the right ways where it'll make me think of the right way to do something, which is nice. So even when it's wrong, it's quite helpful. I think of it like it's easier to edit something than write something completely from scratch. So I really like these tools. So anyone out there who's thinking, oh, is this stuff something you want to be doing, like you just said, just give it a try. It's I mean, ChatGPT is open source, like this. I mean, not open source; it's free. You can get into the beta or whatever it's called. And Hugging Face, you don't pay anything, which is the weirdest thing. Like a lot of these services, like you can run it in Hugging Face. I'm surprised that. I mean, imagine there's some computation or something happening. Like this isn't free. Like nothing on the internet is free. Like someone is paying for that data and that transaction and the compute to happen. So take advantage of it. It's a really great time to be alive to play with these things. And I'm sure there's someone out there who even ChatGPT could probably answer these questions even more. But
I will throw in one more thing. There's been a lot of talk about the availability of Nvidia GPUs and everybody talking about how much compute it needs. A lot of these models are small enough that you can run them, train them, extend them on a normal laptop without any special hardware. It just gets faster from there if you do have a gaming computer or something to run them on. But it's the bar for just starting to get in and dabble is pretty low.
I think I was reading that ChatGPT for number 3.5 costs like 900,000 or something for the data, just like the compute of the data was like 900,000. Which, if you think of the fact that they made like $1 billion, like that's a pretty good return on investment. If you can turn $900,000 was just to compute. Granted, they had a whole team that's probably costing a lot more than $900,000 while that's going on, but it's I feel like that's relatively cost effective. Are you guys building your systems on-prem? Like, do you have like a little supercomputer, or are you doing something in the cloud?
We've gone on and off. So we started our first GPU computing partnered with IBM using the soft layer data centers because in 2015 they were the only game in town for Nvidia GPUs in the cloud. But they had a nice entrepreneur program that they sponsored our first couple of years there, and then we moved to Microsoft Azure with their BizSpark program, and then followed on with some sponsorships from Nvidia to fund the GPUs there for some big projects that we collaborated with the Michael J. Fox Foundation on. And then we hit a point where, we had some predictable compute power for baseline training, and ended up building some on-prem capacity with, like 24 GPUs that we could do basic sets of like training on, and then still keep connection into cloud, so we can do hybrid, kind of a hybrid situation where when we have projects, we can spin up a bunch of nodes on Azure or Google Compute or wherever, and then run our training and then turn them down when the project's done. And then we always have this kind of R&D center on-prem.
That sounds like, as I just imagine like getting to play with that every day. It sounds like a lot of fun. I've built some stuff here, which I won't name, so no one hunts for it on the internet. But because you're only as good as, like, security is only as good as like the neighbor next door's less security, and the best security is knowing that you need security at all. But so I've been building some little small things for fun, from old computers and things that I found on like Facebook Marketplace and stuff, and Raspberry Pis, and so it's fun to see these things be built and play with them every day. Even if you could find, like, there's something really weird about building something on-prem. It feels so good. I don't know about you, but it feels good to me to build these little servers and then see them like light up and work, and you can do like compute stuff on them. I don't know if you like built it all yourself in house, or like you like hired someone in.
Yeah, no, absolutely. I mean, I started tearing things apart when I was a kid, and usually got them back together okay, but then was building my own computers like early on, and continued that through. So I still did that here too, and it's a fun project for the team to go out and like figure out what are the best components and how do we want to organize this. And in this compute environment, it's kind of interesting seeing the big wave of everybody doing compute around like crypto mining a few years ago, which used a lot of the same hardware patterns that are now being used for AI. And a lot of that is pretty applicable to set something up that you want to run these little build at home supercomputers, basically.
I always wonder about like how long does technology last before it either becomes relevant or just degrades and dies, because things aren't built like in the 50s where they last for a long time, especially since like things do just become obsolete. If I was a crypto miner 5 years ago and I went all in on Bitcoin or something, and I have a big farm, because I've been reading about people who did that and then they lost all their money or something, and so they had to close them down. It sounds like there might be like a leasing opportunity or something where they could like make those resources available to someone out there to link into, and then they could be like the on-prem servers for them, which is just, I guess, the cloud in this situation. So is it possible for these things that are 5 years old, these old crypto people, old even though 5 years, to retool themselves for AI, take advantage of it?
I think a lot of the bigger players did custom built ASICs, these purpose built computers that were on silicon, just made to do mining, and those would probably be difficult or impossible to repurpose directly to AI. The hobbyists who had a bunch of GPUs that they were just using, under the bed, those are probably more easy to repurpose, because that Nvidia platform is really built for generic computing. It's just whatever you want to run through it. Yeah. And I think you'll see similar things on the AI side now. In fact, I've heard several companies are starting to design ASICs for AI compute. That the same kind of thing, where with Nvidia GPUs becoming scarce, we now have new people that are making purpose-built hardware that'll be much faster for AI training, and those will probably become just as big for a while. For anybody who's looking to buy shovels and pickaxes right now, or to sell those.
Yeah. It's interesting. So you're aware of the metaphor with gold mining, where the people who make the money are the shovel and pickaxe seller people. Well, a lot of people don't know that one. I don't know why. It's a really good analogy whenever people think about business or how to make money. It's like, don't be the people who need the shovels and pickaxes, be the people who build them or something.
Right.
Yeah.
Although if you look back from that time, the people who built the railroads and banks are the ones who are still around today. So maybe maybe the analogy
That's
isn't
fair.
quite as accurate as people like to. Makes it easy to repeat, but maybe not be all it's corrected for.
Yeah, it sounds like the real lesson is be a bank, own a bank, and then invest in political campaigns so the winds never go against you. That's probably the right thing. I think like no one got locked up. The only people in the entire world in 2008 that got locked up, I think, it was in like Iceland for the 2008 financial collapse. But so we've been talking around this a little bit. Vincere Biosciences, which I believe on your landing page, Vincere is Latin for victory, which is fun. I like Latin as well. There's a lot of Roman history behind me, including some over my right shoulder. But what is it about? And obviously it has AI in it. And there, you know, if anyone's reading the title of the thumbnail, it's going to have biology as well. So could you just tell us a little bit about what it does? What is the purpose of it?
Yeah, our initial idea was to leverage computation and data to find targets that were better for diseases of aging, in particular Parkinson's disease. And using those, we really came to strong conviction that enhancing mitophagy would have potential to stop the progression of Parkinson's disease. Mitophagy is the process of recycling damaged mitochondria. You may remember mitochondria from science class as the powerhouse of the cell, the one thing everybody remembers from school. But they're really a lot more than just the energy producers. They're also nutrient sensors and viral invasion detectors and coordinate a lot of epigenetic changes and cell-to-cell communications, inflammation and other things. And so as people age, one of the hallmarks of aging is that this process of damage control of mitochondria through recycling degrades. You get less efficient ability to recycle your mitochondria. And so with this mechanism, we were like, it would be great if we had a way to boost that so that you could recycle the damaged mitochondria more quickly, bring the cells back to health. There weren't any good, clean, small molecules that could be used as drugs for that. And so we started drug discovery programs and now have a pipeline of small molecules that can increase the rates of mitophagy. They do what they're supposed to in cells, they do what they're supposed to in mice, and now we're preparing them for development into human testing, hopefully within the next year or so if everything keeps going well.
What for the mice studies, what was the result of the drug interventions?
We just actually completed a few of these, which we haven't published the data yet, but are working on using some really cool new tools that just came available in the last few years of transgenic mice that have fluorescent mitochondria. Two different systems, one called MT-Chema that was developed by Nuosan and Torm Finkel at the National Institute of Health, and the other MitoQC developed by Ian Ganley at the University of Dundee in Scotland. And through a recent Michael J. Fox funded project, we were able to license both of these. And the way that they work is they have these fluorescent tags on proteins in the mitochondria, and they normally glow green. But when they go into the acidic environment of the lysosome, they flip to glowing red. And so with this, you can visualize the whole mitochondrial network and where all the mitochondria are and what kind of shape they're in. And then also what proportion of the mitochondria are undergoing mitophagy at any given time. Super cool models that didn't exist 5 years ago.
Do the colors blend? So like when they're in between stages, does it like a mix of the two colors? I don't know what the mix of the two colors would look like.
It's a hard flip at
binary.
2 wavelengths. Yeah. And so they both work in slightly different ways, but the end result is that you get a distinct wavelength of green when they're outside, when they're normal and healthy, and then a distinct red wavelength. And you can measure this with confocal microscope and then using tools like ImageJ or others to quantify the intensities of wavelength. And then there's some other emerging interesting, back to the AI, there's some neat things that you can do with that as far as training models on edge detection of the cells or color detection, how many pixels are one versus the other, and starting to categorize what you see in a healthy versus a diseased cell or a treated versus a not.
So then you have this base of being able to see when it's happening versus when it's not happening, and then you can intervene with your drug and then see very directly what's happening, and even potentially there's like dosing or like how to deliver as well. You could learn with machine learning like over time. That would be something as well. Like you could fine tune it, because some like a lot of drugs are basically like they'll kill you at a certain rate. So it's like what rate does it you need to actually have the where the poison isn't worse than the cure, like what actually is going to help you. So I imagine like that all that's in there as well.
Yeah, that's one of the things with any new chemical. You want to identify what's called the therapeutic index or therapeutic window of what's the lowest dose, that minimal effective dose, and then what's the maximum tolerated dose. And that kind of gives you your safety range that you can work in. And one of the nice things about these molecules is they have very little effect on healthy mitochondria. They don't directly depolarize the mitochondria, and they only shift basal mitophagy by a very little bit. But if the mitochondria become damaged, in the lab, you can use something like CCCP or oligomycin antimycin. These are toxins that damage the mitochondria. When you do that, the cells naturally increase the levels of mitophagy to a certain extent, trying to recover and clean themselves up. But if you add our molecules, you get a really robust dose-dependent increase in mitophagy on top of that. And so with that, we've been able to identify an optimal dosing that can get the cleanup of damage. We've really focused on making sure that the compounds can do this one mechanism really well, more so than looking at disease models, because like mice don't get Parkinson's no matter what you do to them. So I think a lot of drug discovery is focused on trying to demonstrate efficacy in a mouse is kind of misguided, particularly for something like this, where half of the genes that cause mutation or that cause Parkinson's disease are directly within this pathway. So we know that just from human genetics and from human pathology that like if we can get this mechanism right, it's likely to be beneficial. So our focus is to make good, clean drugs. We'll test whether they work or not in humans, not in mice.
Yeah, makes sense. Are you in a state where you feel confident, like teasing a little bit, like how good the intervention happened with the mice turning on the colors and not, or would you rather wait for the studies to come out?
Yeah, no, it's very clean. We've shown a similar effect in the MT chema and the MitoQC mice, and with measuring protein levels using like a Western blot. And those all correlate really well. So we've rigorously shown that we're able to increase mitophagy to the same extent with these different models. So we know what that is. And that level that we see with our drug is similar to the amount that another group, David Simon at Harvard last year at Society for Neuroscience, showed data from USP30 knockout mice. USP30 is the protein that our molecules inhibit. And so if you take that protein completely out of mice, it also raises mitophagy by a similar extent to what we see with our drug in these three different models. So that's kind of an outside validation that what we're seeing is probably about maximal increase in basal mitophagy. He also then showed that those mice are very protected in models of Parkinson's disease. Again, skepticism around mouse models of Parkinson's altogether, but if you use a overexpression of alpha-synuclein, which is the protein that aggregates in Parkinson's, if you do that in the mice that have the USP30 knocked out, they are almost completely spared from neurodegeneration, and they have dramatically lower levels of abnormal alpha-synuclein. So those are the two key things that go wrong in Parkinson's disease, which can be induced in the mouse with this model. So gives us some extra evidence that the mechanism that we believe to be linked to the Parkinson's pathology is protective in one of the aspects of Parkinson's pathology.
Did the mice respond to the intervention in any other ways in terms of like health span or like longevity? Because I know there's some research out there saying that if you could boost up essentially the mitochondria, like you'll have some other types of effects on it. Or was it also like a lot of times I always wonder, like, oh, could you study all these other things at the same time? But usually it's like we're focusing on one thing at a time. So I understand if not.
Yeah, I would love to see that. I don't, not in this particular study, and we haven't done long-term studies to be able to look at lifespan. Those take years and are quite expensive. The mechanism, though, we do believe would have some of those effects. So to step back into the mechanism a little bit more, when mitochondria become damaged, they get tagged with chains of ubiquitin, which then recruits an autophagosome to form around the damaged mitochondria. And then that whole bundle gets carried off to a lysosome where it gets degraded. Those ubiquitin tags are added by a protein called Parkin, which is an E3 ligase. And then in most biological systems, there's a check and a balance, a yin and a yang. The USP30 is the check on that action. It's a deubiquitinating enzyme that is always on the mitochondrial membrane. And so it's just constantly looking for ubiquitin tags to take off. And so it's kind of setting this threshold of, if there has to be a bit of damage in order for Parkin's activity to overrun the activity of USP30. So that's where if we inhibit USP30, we can increase that process. Well, the other way you could do that is by activating Parkin to add those chains more quickly, you get the same effect. So all of that just to introduce Parkin as a player. And in fruit flies, if you overexpress Parkin, if you make it so there's more Parkin, the fruit flies live longer and they're healthy for longer. The females are fertile for longer, in suggesting a increased health span. And then the lifespan is also increased. You get both a health span and lifespan improvement. So we think that should translate to we're hoping that translates to human as well, right? You don't know until you know. But the mechanism seems to carry. One of the things that we see in human that is kind of related to this, the quantities of Parkin go down as people age, even without any disease, just as part of natural aging. Parkin levels decrease. USP30 levels don't seem to decrease. And so I think that's kind of what happens. You end up with this imbalance of the natural homeostasis where you've got less Parkin, but USP30 is still chugging along at the normal levels. And so you end up with reduced mitophagy as you get older. If you inhibit USP30, you restore that balance and you could theoretically get youthful levels of mitophagy, even though you've got less Parkin.
Do we know why one degrades and one stays the same? I mean, doesn't degrade at the same rate? Or are they both degrading and the one just like Parkinson's just degrading faster? Or is
That's
30 just the same? Yeah. Do we know that?
Yeah, I don't know. That would be a good one to graph over time. I think they should pull all of those data back together and look at that. But I don't know the answer to that, whether they both go down and just one's going down faster. Or why physiologically, why one would degrade over time and the other wouldn't, or even why the one degrades. Like the why of aging is a really interesting philosophical question.
Yeah. Well, what if Parkin's going down and you can decrease 30 to match the level, could you also, is there any downside to increasing Parkin to keep it at that level versus decreasing one to a different level?
No, it's actually a really interesting approach, and that's one we've been working on for a while, and a couple other groups have been trying. Overexpressing proteins, you can do that with gene therapies, maybe, or but it's generally there. You introduce a whole lot of like unknowns when you with those new modalities of drugs. Small molecules like typical medicine type drugs, it's very difficult to increase the activity of an enzyme. Inhibiting things is really easy. You stick something to a protein and it usually loses its function. But trying to make something more active is difficult from a chemistry perspective. That said, we actually think we have some Parkin activators in hand now. We're just starting to characterize them in cell models, so they're a little bit earlier. But so far, they seem to increase the enzymatic activity and to increase activity, the rates of mitophagy in cells. There are other considerations as well in that the way this process works, Parkins, that trigger that when there's damage, it adds things. So it's kind of like the gas pedal to the system. And if you push down too hard on the gas pedal, you might go really fast and you can take away your healthy mitochondria. Whereas if you're taking your foot off the brake, there's only a natural limit to how fast you can go. You can't like run away too fast just by taking your foot off the brake. So from a safety perspective, I think USP30 seems like it's kind of a safer path to this outcome if you can get sufficient increase. Whereas Parkin, you might project that there might be a kind of more narrow safety window with that. So there's some considerations in terms of like the different approaches. Yeah, we're certainly interested in that approach and continuing to pursue that ourselves.
Yeah, it makes sense. Reduce the complexity, makes it easier, especially with all the other things you have to worry about: funding, employees, all these other different variables. The simpler you can make the innovation, in reference to what we're talking about, sounds like the better route to go down. What do you guys expect to be the effect? If we're focusing on Parkinson's as the age-related disease, what effect do you guys hope? You're not at the level where you can make the determination with the mice studies yet, but what do you guys hope will be the impact? Like, will it be something that can help Michael J. Fox, for instance, or for someone who already has it pretty significantly, or is it someone that has early signs and you hit it before it gets too advanced? Like, what is it going to look like?
Yeah, I think in all cases, the earlier you can intervene, the better, because this is a progressive neurodegenerative disease. So the population of remaining healthy cells that can make their connections, matters. And it's unlikely that this type of intervention would cause new neurons to come into the space. But what it can do, we believe it should stop the neurodegeneration of these cells. The particular types of cells that are primarily underlying the motor symptoms of Parkinson's are these substantia nigra dopamine neurons that are known to be very susceptible to mitochondrial toxins. And so because of that, we think that this should give them the protection that they're lacking to these mitochondrial toxins and stop the neurodegeneration. So you would stop losing more neurons. And if you can hold those neurons as healthy, then the natural synaptic plasticity, neurons are really good at like going out and making new connections. So if you can stop the ones that are left from dying, the ones that are there will then go back and regrow. And we see this in other types of brain injury and things where like incredible ability to rewire, to have the remaining parts of the brain take over for the parts that have been damaged and get back normal function. So we believe that this intervention should stop the neurodegeneration, which should then allow the brain to recover. So then progressively getting, better function as well. So symptoms would improve over time. Neurodegeneration would stop very quickly.
So it is something that could help out Michael J. Fox as well as it's the whole gamut. It's not, for instance, geared towards people with late stage or any stage in particular. It looks like it'd be able to help everybody and have an effect no matter what level. Yeah. Yeah,
I think help across the board. It's certainly hopeful to show benefit for everybody. we're definitely hopeful that it would have help there. Also, speaking of Michael J. Fox, the Fox Foundation has some really exciting initiatives that have just been announced this year with early testing. They've identified, with help of other researchers, there are some abnormalities in people's ability to smell that are one of the earliest signs of Parkinson's disease before any tremors or motor symptoms are detectable. And those correlate also with an available test for the levels of aggregated alpha-synuclein protein. Right now, looking in cerebral spinal fluid, but other people are working on translating that now to a blood test as well. And so with these combination of these tests, the Fox Foundation is really hopeful that we can start to detect Parkinson's at its very earliest stages before any symptoms come. And coupling that with this type of intervention, we may be able to find the patients in need before they ever have their first tremors and prevent Parkinson's, like clinical Parkinson's, from ever having, from ever starting in the first place.
Yeah, that's a beautiful vision. I hope the clinical trials come soon, and then you guys can get something out there as soon as possible, because people definitely need it. I mean, there's people that might get those results right now, today, or this year, and I hope everything works out in terms of the clinical trials and stuff, and I hope everyone listening as well. Is this going to be? Is this like a shot? Is this like a pill? Like how? So, and then like how often will people have to take this? And then
we're working toward a.
Yeah, go ahead.
I was gonna say we're working toward a once daily pill. That's the goal.
Okay. So if I'm, and what's the average age for people who develop Parkinson's? Is it like 35 or 45? Like how old do you be? How old do you typically? Yeah.
Yeah, the average age of onset is in the early 60s for most patients.
Okay.
And most don't have any genetic mutation or cause. They're what are called idiopathic or sporadic Parkinson's that comes on in your early 60s and you don't really know why, and then it goes. There are subtypes that are called early onset Parkinson's or young onset Parkinson's that are more often genetically linked with mutations in PINK1 and Parkin right in this pathway. And those can onset in 30s or even younger sometimes.
Your therapy will be able to affect those as well, just universally?
Yeah, we think all it has potential for all, given some of the modeling that we've done. We think it will work for the sporadic Parkinson's. It's a clear link for the patients who have mutations in this pathway. Those early onset Parkinson are probably the ones that we would look at first, because they have deficits right in this pathway. The other side to that in consideration, though, is the early onset Parkinson's tend to progress much more slowly. So that means clinical trials for those patients have to be longer to confirm the effect. The benefit is there.
Yeah, that makes sense.
A lot of considerations as you go into clinical trial design.
Yeah. Do you expect, does the science back up that this would be good for a person's entire life? So it'd be a pill they take for the rest of their lives? Or is there any research that suggests that the body would start resisting or become immune to it?
We don't know those. I mean, the expectation would be that it's one that you would have to keep taking, or the imbalance would go back to where it was. But we really wouldn't know if there are other adaptations until we do longer studies. This is a mechanism that has never been drugged before, which is kind of an exciting place. A lot of drugs that go through the clinic are just targeting the same mechanism in a different way. But there's never been a drug that has worked on USP30 or been able to increase mitophagy cleanly. There are some natural substances that increase mitophagy, but nobody's really sure exactly how they increase mitophagy, and some of them actually look like they're maybe mitochondrial toxins at high doses. You have to be a little careful with some of those supplement type thing.
Yeah. So then it's 2023, clinical trials within a year, ideally. What stands in the way from today to clinical trials? Is there like FDA needs to give you need to give the FDA certain data? Like where are you from that? And then part two of the question is the now until like when someone would expect to be able to use it if everything works out well.
It's a great question. So standing in the way of first human trial, there's a battery of formulation and efficacy tests, or safety testing primarily, where we have to demonstrate that, identify how much of, at what level does this drug become toxic? Everything's toxic at some level, and you have to find what that maximum tolerated dose is. You have to do that in a couple of different animal species, a small and a large, so that you can project what the toxic dose for a human would be, and you need a couple of those points to extrapolate from. So going through all of that data then gets submitted to the FDA in what's called an investigational new drug package, where we'll go through a review with their committee members to look at all of the data and kind of get that stamp of approval that we have a hypothesis that is sufficiently justified to test in humans, and that we've demonstrated sufficiently that the molecule is safe enough to do that first test. With all of that, then we would be able to start the phase 1 clinical trials, which really are specifically looking at safety. So in that you'll test in 50 to 100 healthy volunteers who will take a single dose and then multiple doses of the drug, and just look to see if there are any adverse events. If there are no adverse events, then you're good. Ideally, you'll also somehow measure a marker to show that the molecule has engaged the target. We want to know that our drug not only is safe, but it actually inhibits USB30 or increases mitophagy in humans during that first trial. So that's kind of that first battery of work, where we're about a year from being able to do the IND, probably another year to have the final data analysis and readout and everything on that safety in humans. From there, for Parkinson's disease, the efficacy testing, where you do a phase 2, which is kind of a small number of patients to test proof of concept, followed by a phase 3, where you do a large number of patients to really demonstrate true efficacy over a durable time. Those take 10 years or so. The phase 2 is maybe going to be a year or two. Phase 3s are in the realm of 10 years. So altogether, you're talking 15 years before this is approved and ready to go to the patients. And if there's some of these stages, there's no technological way to speed this up. Like, maybe we can get a biomarker, but to show the benefit faster, and we can, declare success. But really, it's such a slow progressing disease. You need to show that long-term benefit. One of the things we're also looking at is using the molecule for other diseases in parallel with Parkinson's disease, because we know mitochondria are everywhere in the body and are important for other energetically demanding tissue. We may be able to use this in a clinical trial for something like chronic kidney disease or heart failure, where you have potentially a very short clinical trial, and we could do that phase 2, phase 3 efficacy piece in a year or two. So we may be able to get a drug approved in the realm of 5 years as opposed to 15, taking it forward for one of those approaches in parallel.
Yeah, I was wondering if there was like an orphan drug status or emergency pathway or compassionate reasons that would allow someone to, sign a waiver saying this is experimentatory at a certain stage of, not stage 1, but like later stages, where they could have it sooner if the need was greater, if it would expedite it in any way. How do you I don't know, how do you like stick with it when you're looking at something that you're already like several years into and you got potentially 5 to 15 years ahead of you before someone can really smile knowing that they're going to be using that and do better from it. How do you just keep grinding every day?
Yeah, those are a couple of good questions. First, to address the rare disease piece, it's something we're definitely looking at. The
Mm,
one benchmark that I kind of keep asking myself and asking the team is, what's the shortest path to get a molecule that you would be comfortable taking yourself, that I would be comfortable taking
mm,
myself? And I think some of that basic safety testing is a requirement that like we just have to cross that hurdle to show that this is something that like, yeah, I'd feel good starting to take this molecule and see if it does something good. We have to cross that before we can do any sort of compassionate use or trial type things. Because there's that risk to reward balance. Once you know
mhm,
it's reasonably safe, then you can start to look at the reward side. Uh, and I think once we have that achieved, there are a few options for accelerated approval for some of those like genetic forms of Parkinson's that are smaller groups. There are also rare kidney diseases and some of these heart failure type things that can be, I think, could fit an accelerated approval path. So we're certainly considering that as well. To the broader question of how do you maintain the faith through this
mhm,
long process, the biopharma world really, in a lot of ways, is a relay race toward the clinic. And so we've built this really great early discovery engine where we can come up with novel hypotheses and do medicinal chemistry and make molecules and get them to the clinic. It's likely we're going to hand off the baton at some point here through a pharma partnership to
mm,
one of the pharmas who do a lot of clinical development and can execute those larger trials more effectively. And we'll continue to partner and work with them. But I think a lot of the execution then moves to that bigger group who has a lot of clinical expertise. And then even then that may hand off another time to an even bigger pharma that has worldwide distribution and manufacturing capabilities and marketing and all of the things that you have to do to give a drug to tens of millions of patients around the world. So those different stages of complexity and different types of jobs you have to do, like the manufacturing and
mhm.
sales and marketing is complex, but entirely different type of complex than figuring out what you should make a drug for and then optimizing that molecule to get it in the clinic.
That makes sense. So speaking on Michael J. Fox Foundation, uh, what did that look like? Are they basically just like an NIH, like a little mini NIH, and they give out grants to people, or is there like a more in-depth partnership with them?
It's definitely a more in-depth partnership than the NIH, and both are great organizations in different ways. But the Michael J. Fox Foundation is really special. I think Parkinson's is much more imminently likely to be solved because of the Fox Foundation.
Wow.
They not only give out grants, but they also give access to data, and they very strongly encourage multi-institutional collaborations. So we're members of research consortia where every week or two we're on a call with the pretty much the entire core Parkinson's disease research community, sharing data and talking about what is going on. And that's academics to other competing biotech companies in similar spaces. Like we're all working together to share information to push the science forward because the goal is to get something that can stop Parkinson's disease. They on the data side, they have this Parkinson's progression marker initiative, which is biomarker data from patients that
Hmm.
is like nothing else in the world. And we've been able to import over 50 terabytes of patient data that include whole genome sequencing, RNA-seq transcriptomics, and DNA methylation data from the same human participants, all correlated together, tracked every 6 months over multiple
Wow.
years. Like such a wealth of data that they've invested in building, which we can now build regression models that can start to predict the biomarkers of progression, or classification models where we can start to subtype, which patients are more likely to have a response to a mitochondrial therapeutic, and maybe which ones are more likely to benefit from a lysosomal therapeutic, and which ones are more likely to really just benefit from a synuclein-based aggregation approach. And so that data is helping the field now to define more precise clinical trials. And the more precise we get, the less noisy the trial results, and the more likely we are to do accelerated approvals and all of these other things that, like, we want to show definitively that any intervention does what we said it was going to do, and that it's protective for some subset of patients, like as quickly as possible, right? And so Fox Foundation, through their data and their funding and their community building, really have just put Parkinson's disease on a different playing field than Alzheimer's or any of the other neurodegenerative spaces.
Yeah, I was thinking about Alzheimer's, and there's the Alzheimer's Association or something, but I don't think they do biobanking or any of these types of things. I haven't heard of another organization having all, like, the community, the data sharing, all these different elements of it. That's really powerful.
Yeah,
And
I think a lot there's a lot that are focused on patient advocacy, which is also clearly important and benefits there. And a lot of these others do. There's a ton of research. The National Institute of Aging funds a lot of Alzheimer's research, and there are brain banks that are collecting patient samples related to Alzheimer's, but it's much more dispersed.
Yeah, it's not aggregated.
Yeah.
It's an aggregation of that stuff that's really powerful, especially as we're getting these machine learning models and this AI models. And everyone's talking about, like, how deep is your data lake? It sounds like Michael J. Fox, the foundation, has the deepest data lake for Parkinson's and potentially age-related other illnesses, as they've been, tracking people for every 6 months. And if there's lots and lots of people that they've been tracking, that's massive. Like, no wonder, like you're saying, it feels like it's imminent in terms of, like, a cure or at the very more like a treatment to help people. So would you think that end of 2030s, like, there could be something where people could, if they got the diagnosis, like, hey, you have Parkinson's, that there's going to be things there for them to help them so they don't suffer? It's not like a slow, like a down to the right versus up to the right. They can just keep having healthy life.
Yeah, I think that's plausible in that time frame. And I mean, caveat that with an acknowledgement that having the perspective of having been tangential to the Parkinson's field for over 20 years, that in the 90s, when genetic mutations were found, everybody thought Parkinson's was imminently solved. And in the 80s, when some toxins were found to cause Parkinson's, everybody thought it was imminently solved. So, it's not done till it's done. But it feels like this confluence of data availability with AI tools that can make sense of that data, with new diagnostics that can detect earlier, and new interventions going into the clinic that are working via distinct mechanisms. I think we've got a lot of shots on goal that will come to fruition during that time frame. So I'm hopeful.
Yeah. Also, learning from the past failures. Every failure is an opportunity to learn. So going down these different quagmires, potentially, there's a lot of contextual subtext of things that you can learn there that then influences how you think about a question or a problem now, even structuring it. So even although I'm just saying, which is to say that not time wasted, I imagine that we still learned a lot from those time periods that have helped accelerate what's going on now. Even if it's just
absolutely.
like, we know not to go down those routes. Yeah.
Yeah. All of these clues, it's, we stand on shoulders of giants, and, everybody that has come before building the information. That's a really exciting time to be alive right now. We see exponential growth on biological methods and data production, as well as compute. And it feels like everything is just accelerating at an accelerating rate. It's a fun time to be around.
Yeah, it's like sometimes I feel, is it a bittersweet time to be alive if you were to be diagnosed with something right now? Because it's sweet in the sense of there's so many people working on these problems. There's so many great tools coming out. There must be something coming, and it's bitter because you're going through something and there's not necessarily a great treatment for certain things right now. And so you live at that precipice of like, it depending on how you progress, like you may be fine because then there'll be a drug that comes in and takes care of you, or maybe you'll be like the last generation that falls to it. And so it's like it's a great time to be alive, even in the sense that like there at least is hope. But then sometimes I wonder, is it bittersweet? I imagine myself as someone, if I was diagnosed with Parkinson's, like, would I feel that way? It's like, well, there's a lot of hope. There's something to hope for, and I think that any time in history, like, there's a lot more, like you're saying, like shots on goal to suggest that the people could be hopeful, and I think that's a powerful thing in of itself. Just to, if you're trying to get up, exercise, do all the different things, like can slow the progression. I believe I was reading that exercise can have some type of impact, just aerobic exercise or something, in terms of like making sure things are staying connected and whatnot. So if you had no hope and it was just like, oh, you'll suffer less, which is still useful, and but there's hope that you do these things. I imagine that makes it easier to get out of bed and keep fighting, as you and other people keep fighting on your side of the party to deliver deliver something to people. So like bittersweet, but still probably the best time, like you're saying. The transitioning to looking at the time we have left, transitioning to books real quick. On Twitter, you said that The Creative Act from Rick Rubin is a great book. Why is it a great book? Why would you recommend it? I have not read this. This is not a challenge. But what about it do you find significant? And then I'll ask you a separate question.
Yeah, I'm at the early stages of the book, but so far it has exceeded expectations. So, I mean, Rick Rubin, if you're not familiar, is a music producer who has spanned like every genre, Def Jam Records and BC Boys and like some rock, country, to everything, and clearly has an eye and an ear for what's interesting and entertaining. And so his perspectives on creativity, I thought would be could be beneficial. And I'm actually listening to the audiobook, which he narrates himself, and it's almost like a soothing mantra of little tidbits of facts and tips, and it just keeps going more and more interesting into the way the universe is connected and the way creativity sprouts, and ideas happen because their time has come. And like, it's why you often see many people will create similar things at the same time, and how you can tune yourself into that more and open yourself up to be aware of what is happening around you, to actually experience the world, to cut out some of the noise, and then to just allow yourself to receive what the universe is handing you, and then be a filter to that, to think about, like, we have all of this input that's happening around us, and our job as creators is to be a filter for that, to allow the right spark of our thing to come back out in a unique way that then is pleasing to other people, right? And if you think about it in that way, and it's one of the things it says, like, all of us are creators, like, just in how we chose to make this sentence, or to get out of bed, or which path to take to work today. These are all creative acts. And so these tips of creativity can be applied to everyone, whether you're trying to top the charts, or create new drugs, or just have a conversation.
I'm gonna have to check that out. That sounds amazing. Especially given the fact that I have to ask questions of people all the time and digest and be creative. So, are there other books you recommend? Or
yeah, I mean, as you were talking about the hope a minute ago, that reminded me, a while back I read How to Live Long Enough to Live Forever by Ray Kurzweil,
Hmm.
who's a futurist science inventor kind of person. Anyway, in this book, he lays out this idea, that at some point there will be this singularity where humans and computers will merge, and we can upload our consciousness and live forever in this silicon substrate, and for all of eternity, which sounds insane. But then he says, in order for him, as somebody who's getting up in age, to make it to that point, he has to find bridges to get there. And so he started with this idea that, like, okay, by optimizing my diet, sleep, and exercise, I can increase my odds of living to 100 instead of 80. And then by adding a really unique blend of biomarker tracking and supplements, maybe I can increase that to 110 or 120. And maybe, just maybe, by the time I hit 110 or 120, these biological reprogramming companies will get their stuff together, and we'll be able to turn back the clock. We can already in the lab take skin cells and turn them back into stem cells, and then take them forward into heart cells and whatever. So that next step is we're going to be able to take our skin cells and generate a replacement heart, a replacement kidney, a replacement liver, and we can be our own transplant donors in sometime in the next 10, 50, 100 years. And then from there, do that at a full organism level. So I think that was a super interesting book if you're into like this futurism and longevity and health. That one impacted me a lot to think about, like, okay, how can I change my life to try to maximize my odds of living forever, hitting that longevity escape velocity, as people talk about. We don't know when it's going to happen. It may not happen, but man, I sure feel healthier the way I'm living my life after making those adjustments anyway. So if I'm healthier and more productive along the way, it's kind of a win-win.
Yeah, exactly. Sometimes people don't take care of their body, but when you take care of your body, you're making your brain much more have more horsepower type of thing. So definitely agree there. So we queued a bunch of people for questions for this, and one just came in and I got an alert. So I was like, oh, okay, I've never read one at like literally 55 seconds. It just came in. So this will be kind of fun. So and everyone has fun sign-on names for some of the social media. So Salt and Pepper 59 asks, and I have not read this before saying this, so hopefully it's not egregious, but one could imagine that excessively boosting mitophagy could result in the removal of healthy mitochondria, which could have a toxic consequence. Are there any indications from in vivo studies in healthy animals that could be that could prove that? Is there? Yeah, okay, you probably understood the question. Go ahead.
Yeah, it's certainly a concern. Looking at mitophagy as a target, we want to make sure we leave the healthy ones alone. That is one of the things that we're in a unique position, having Parkin activators and USB30 inhibitors that we can test. And some of our early data there do suggest that USB30 has a much better safety profile. We're still working through, fully analyzing what that means, but that's one of the reasons we've prioritized pushing USB30 forward as a target, is because we do have repeatable, conclusive data showing that it doesn't clear the healthy mitochondria. When mitochondria are good, they're pretty much left alone. And then when you have damage, it kicks into overdrive.
Sweet. So Salt and Pepper, hope that answered your question. I think it did, if I were imagining from your point of view. So another one, and I'm going to call them Kane because I don't know the appropriate. I think this is their real name, so I'm not going to say the whole thing. So Kane's asks how to skip from P1 to P3 utilization regulatory grade RWD with oncology breakthrough designation as a lens. I don't understand that question, so hopefully you do.
Right. So to interpret that, what I think is being asked there is, how could you fast track from a phase 1 clinical trial drug to a phase 3 clinical trial drug, and then start tracking real world evidence of the benefit and using the breakthrough designation? And this comes back to something that you were asking about earlier as well, of like, how do you get the compassionate use or get more people doing this. I think when you get to efficacy testing, I do think it's really interesting that like you want to track for a long time. At some point, if your drug works, maybe you run into more ethical considerations of having to do a placebo-controlled trial for a 10-year period, as opposed to letting that placebo group take the drug. And so the way a lot of the trials are set up is, the placebo effect on drugs is really high. So you definitely need to do a placebo-controlled trial to demonstrate that the drug is better than sugar pill. What most of the trials are designed for is a paradigm where you'll start with a placebo-controlled group and do that for some period of time and track, and then maybe flip the groups, or have your placebo group will roll into a treatment group in a new cohort. And so then you can start to see this group that was on placebo, then got on drug, and compare like placebo to control, and then placebo, once they got on drug, did they also catch up? Or, how durable, if you take these people, the first drug group, and flip them back to placebo, do they then drop off, or does the benefit stay durable, right? So there are interesting things you can learn by flip-flopping your groups. And so I think you need to do that to conclusively know that your drug does have a beneficial effect. That said, if you get to real world evidence and you know your drug is safe and you just start giving it to everybody, and you can actually track everybody's usage in the real world and see, do they get benefit, then you should have high enough statistical numbers that you can overcome you'll have enough power to see a benefit even without a placebo group. And maybe you can compare to the other, like if you've got 10 million Parkinson's patients in the world, not all 10 million are going to take it, right? So even if you have some volunteer group, you can compare that to the rest. All of that opens up the need to track biomarkers and to be able to detect the quantity of drug in your participants in some automated way. That's a big challenge in clinical trials right now. So let's say you open up this real world, anybody can take it if they want it. How do you know whether they actually took it every day or took it every other day or skipped a week, or, and how are they going to come back for blood testing to show benefit? Are they going to just maybe take a survey every day, every week, every year? So a lot of those, how you go about capturing the real world evidence and the other benefits is there are a lot of questions there, and there aren't really good methods for those types of real world tracking. I'm really curious to see some of these things. Like, I don't know if you've seen Brian Johnson and the
blueprint,
like the blueprint for longevity. Like some of the things, a lot of hype and craziness around the way all that's being done and tracked and shown, but the underlying approach of like, let's measure everything we can about a person and then get as many people as we can to start tracking that same set of things about themselves consistently over time. That now starts to grow a cohort of people who you do know enough about their ongoing health that you could maybe start to try some interventions, like particularly for like a longevity type thing, which mitochondria enhancement might be. That would be a great cohort to then say, we have this now experimental drug that's been shown safe for experimental use. You're already tracking everything. Add this to your repertoire and see if it has benefit to any of your biomarkers. But you've got to have that window of data, which is very few people have right now.
I wonder if you made let's say that someone had a drug and they were trying to do, made the world their petri dish in this way, that if they made the ability to take the next drug or the next prescription, you had to like take a blood test or fill out some stuff, and then you were able to release it, that'd probably make the carrot and stick quite close together, so you'd probably get it that way. That's the only way I could think about how do you incentivize people to know if they're taking it, et cetera, is if it was linked in that way. That'd probably be the only way I think about it. And I mean, that's if you would even be allowed to do it. But for people who've been listening in for the last I don't know how long we've been going, but and they're probably excited to hear where you guys are going to be for the next year to 15 years, depending on how things go. Hopefully not 15. I'm hoping for 5. But what's the best way to stay up to date with what you guys are working on? Is it like a Twitter? Is it a newsletter?
Yeah, we're pretty active on LinkedIn and Twitter. Those are the two primary places. We've also recently set up a YouTube channel and have some cool content there. Spring's been doing these interviews with other people in the field that have turned out to be pretty interesting. So there's a video with the creators of both of those fluorescent mitochondria mice that I talked about. So those conversations are interesting. There's a nice explainer on mitophagy and how the proteins I talked about work. So we'll continue to add content there. And then vincerebio.com is our website, and we're also continuing to expand content there as well.
Sweet. Then I want to thank you for coming on the show today, educating us on the world of Parkinson's and going down the nerdy rabbit hole. Sometimes people don't say the proteins and stuff, or they hand wave it, but you got right in there, and I appreciate that. So thank you so much for coming on the show today.
Yeah, thanks, Lowell. This was a great conversation.